Hello, ladies and gentlemen. My name is Robert Landewe. I'm having a pleasure to talk to you about continuing versus withdrawing biologicals in axial spondyloarthritis. I am a rheumatologist working in the Netherlands. I'm a professor in rheumatology, and I'm regularly seeing patients in clinical practice just as you do. So let's talk a bit about continuing versus withdrawing biologicals. First of all, I'll show you a couple of potential conflicts of interest. I'm EULAR's chair of quality of care. I have my own company for consultancy with different pharmaceutical companies. And I'm also an acting executive committee member of the ASAS organization, which is an organization with which you are probably familiar. So the question is to stop or not to stop. And we are talking about patients with axial spondyloarthritis who are in sustained remission. And the first question that comes up is, why is that patient in sustained remission? Obviously, the most common answer to that question will be, well, that is due to drug treatment. But not so long ago, we also talked about, well, is axial spondyloarthritis truly a chronic disease, a chronic inflammatory disease, or will spontaneous remissions also occur? Or maybe even has a wrong diagnosis been made? A diagnosis of axial SpA is not extremely simple, and people make many mistakes, especially if they confuse classification criteria and use them for diagnostic processes. So that was not only an issue in the ASAS organization, but also at the regulatory organizations like EMA and FDA, when they had to decide about whether or not approving biological drugs for this particular condition. So that is why, especially the European registry authorities had issued a post-approval commitment. That means that the industries who brought a certain drug on the market were approved to do so, but only under the condition that they would do further studies in order to see whether the drug could be withdrawn, whether they were not talking about too much about spontaneous remission, essentially whether it was safe and effective to continue these drugs in patients with axial spondyloarthritis. The question always is, how should you investigate such a kind of thing? How should you investigate whether withdrawing a particular effective drug makes sense or should be done or could be done? Well, there was actually a three-phased process that was developed and that was pursued by many of the pharmaceutical industries with drugs on the market. The first was about selection of the appropriate patients. And when we are talking about axial spondyloarthritis, we definitely want patients with real axial spondyloarthritis, full stop. That means active disease that should be suppressed by treatment. It also means objective signs of inflammation like CRP and MRI. I will come back to that later. And then obviously those patients should be treated in an open manner in phase 1 with the drug in question, for instance, the TNF inhibitor, for a certain period of time in order to induce remission. And when sustained remission was introduced, it was thought, then you could go into phase 2, in which you actually have made a selection of patients in sustained remission, and you randomize them towards either blinded continuation or to blinded withdrawal or blinded dose reduction. This is essentially the template of the withdrawal studies that were pursued in the recent 5 to 10 years. So this was the first one, Ability 3, this study that was done under auspices of AbbVie, who had to do this study in order to prove that the drug was indeed efficacious and effective in the context of non-radiographic axial SpA. And because it was the first study, it was published in the Lancet, it was the so-called first withdrawal study. What was still not on the retina was, and in those days, was whether non-radiographic axial SpA was either a self-limiting disease or even was a disease. So people were very skeptical in those early days. And what AbbVie did was the following study design. So after screening, in which patients had to fulfill classification criteria for non-radiographic axial SpA, they were treated with active drug for a period of 28 weeks, and those who were in sustained remission were randomized towards either continuation of active drug or to stopping. So essentially, they continued with placebo. And the outcome of this study, after 48 weeks of treatment, that meant active treatment or active treatment followed by placebo, the main result could be summarized into this bar graph. It actually led to approximately 80% of patients who remained without a flare in the dose continuing group, so those still on active treatment, and to approximately 50% of patients who remained disease flare, free of disease flare in the placebo group. So essentially, approximately 50% of the patients could be stopped by using placebo, could stop active drug without perceiving a flare of a disease. And that gave us a little bit of the feeling of, is the glass half full or half empty? Although the difference between both groups was highly statistically significant, and it was sold as if you should continue drug because that on average was better for the patient, we didn't lose the idea that in those who were introduced remission for a period of 28 weeks with active drug, that in up to 50% of them, you could safely stop with the drug after 28 weeks and after achieving sustained remission. So what does that mean? How to explain this somewhat strange results? And there's many potential causes. I will mention a few. For instance, many of those patients did not really have axial spondyloarthritis, or maybe TNF inhibition really cures non-radiographic axial spondyloarthritis, or perhaps non-radiographic axial SpA is sometimes indeed a self-limiting disease. We didn't know. Potentially many of these factors occurred in some sort of combination. It was simply not sure how to explain this somewhat strange result. It was not specific for TNF inhibitors, so to say, because we did essentially the same study with our second, I believe, interleukin-17 inhibitor here, ixekizumab, and the results have been published not so long ago. And you saw essentially the same result. Continuing effective drug led to remaining flare-free in 83% of the cases, and that was a little bit more than 50% in the placebo group. So essentially the same result. Is the glass half full or half empty? This was about totally stopping drug. So it was either active drug or completely stopping and continue with placebo. But there was a way in between, and I will briefly discuss with you the results of this particular study, the certolizumab study or the C-optimized trial, because for the first time it was tried to not fully stop the active drug certolizumab, but to provide half dose after a certain period of introduction of remission. C-optimize was a study in which patients with radiographic and non-radiographic axial spondyloarthritis were combined. Ability 3, for instance, the first study that I showed you, was a study of only non-radiographic axial SpA patients. These had either radiographic or non-radiographic. And that means that they had to be either imaging positive or they should have a positive CRP. Imaging positive means a positive MRI, or an abnormal X-ray on the basis of modified New York criteria, or the CRP should be increased, or both. And the induction period in order to obtain remission was not 28 weeks like in Ability 3, but 48 weeks. So it was a far longer period. Keep that in mind. Here is the result of C-optimized. There were three groups essentially. The first is continuing unchanged. So again, these are patients that were brought into sustained remission, and then either they continued, first bar, or they continued half dose, second bar, or they stopped completely, placebo. And there's two things to remark on this particular outcome. The first is obviously that the result, the treatment effect, so to say, between continuing unchanged and placebo or stopping was far more marked than in Ability 3. It was only 20% of the placebo patients who remained flare-free. And I think that can be explained by the fact that those patients were selected more carefully. They had more inflammatory disease. They had run in for a longer time on active drug before they stopped. So you could say, well, these placebo patients, these patients who were randomized towards placebo had a far higher a priori chance to getting a flare because of the fact they had more inflammation. The second thing that is remarkable here is essentially that once you have brought a patient into remission, apparently you can safely reduce the dose by half, because the difference between 83% flare-free and 79% flare-free is actually negligible. It isn't a difference. So half dose apparently also works. In that context, I also want to introduce to you this somewhat remarkable study that was designed in order to investigate whether non-radiographic axial spondyloarthritis was essentially a self-limiting disease. You may think, well, that is a strange question. Indeed it is, but in those days, in the early days, especially in the United States, people were sort of convinced that patients with non-radiographic axial SPA had a condition that was self-limiting in nature. Everything should go away by itself. And that was an important reason for a long time for the drug regulation authorities, the FDA, to not approve TNF inhibiting drugs for the indication of non-radiographic axial SPA, although they had done that for radiographic axial spondyloarthritis or axial or ankylosing spondylitis. So in order to investigate that, they mandated a 52-week randomized placebo-controlled trial of active drug versus placebo, so to say. And that was actually very strange, because for no other inflammatory condition they had ever mandated that in the past. And the longest period that is allowable nowadays for placebo is approximately 3 to 4 months. And this was 52 weeks. And we were, as ASA's organization, very much against this, but they were insisting, and finally we have collaborated in that particular study. It included patients with a diagnosis of non-radiographic, and the modified New York positive ankylosing spondylitis was obviously excluded because there was already an indication for TNF inhibitors in that population. These patients had to fulfill ASA's criteria, and importantly, they either were MRI positive or CRP positive. So in other words, they had at least one sign of objective inflammation. And then those patients were told that if things were not going well, if they had a flare or didn't respond or whatever, they were allowed during the trial to escape to active drug, to certolizumab. And that is important to keep in mind, because I will show you a few important results of this study. To begin with the trial's flow chart. And you are not supposed to be able to read this, but I will pick out a few remarkable things. First of all, in order to get the study popularized, to get the study full of patients, we had to screen 3 times as many patients in order to find 317 that fulfilled the criteria. And that was because it was essentially extremely difficult to make an appropriate diagnosis of axial spondyloarthritis. Apparently, many physicians in the environment thought that the patient was eligible to a study with non-radiographic axial SPA, whereas the criteria thought that was not the case. Most of the these cases of screening failures were due to the absence of imaging, the absence of a positive CRP, the absence essentially of objective signs of inflammation. And then if you look at the numbers completing the trial, you saw that that percentage was far higher in the patients who had received active treatment as compared to those who had received placebo. But the most important outcome of this study, in my regard at least, in my opinion at least, is the fact that in the active treatment group, so in the group of patients who received certolizumab, only 13% shifted to open label certolizumab, whereas this figure was 61% in the placebo group. So That is actually a very important finding in order to overrule the thought that non-radiographic axial SPA with positive MRI and CRP positive, that is a self-limiting disease. Note that this is after 52 weeks. And you see that's also reflected in the major outcomes, ASDAS major improvement, which is a high treatment contrast, or ASDAS 40 response, which is a sort of technical outcome measure that is used in most of the clinical trials in axial spondyloarthritis. This is an unambiguous difference, an unambiguous treatment effect, and no one will doubt these figures. So the drug is effective, also in a 1-year placebo-controlled study, far more effective than placebo. That in itself is hardly effective. Non-radiographic is not a self-limiting disease, to summarize, as long as the diagnosis is correct, and it is a TNF inhibitor responsive inflammatory disease. The majority of patients actually respond well to TNF inhibitor, but signs of objective inflammation are indeed important in order to obtain a proper TNF response. And this altogether was enough to make the FDA approve the indication non-radiographic axial spondyloarthritis and the efficacy and safety of TNF inhibitors. So from that time on, also in the United States, you can prescribe TNF inhibitors because it is effective in axial spondyloarthritis, in non-radiographic axial spondyloarthritis. So what to learn from all this information? First of all, withdrawal trials that you now see coming from almost every company, because they all get the same post-approval commitment, likely often contain patients that actually do not have the disease, but do have pain and back pain and signs of the disease, but not truly inflammatory disease. More selective trials give more interpretable results. Real non-radiographic axial SpA is a real inflammatory condition that is responsive to biologicals. If you have a patient that is in remission, in sustained remission after the start of a biological drug, it's better to reduce rather than to stop the entire dose. In case of non-response, you should reconsider the diagnosis of axial spondyloarthritis. So it's all about what is real axial spondyloarthritis. Well, that is not the same as fulfilling the ASAS classification criteria. It's also not the same as finding a patient who is HLA-B27 positive or have inflammatory back pain. That is rather aspecific. It's also not only those who are imaging positive pelvic X-ray or a positive MRI. There's a lot of misdiagnosis by imaging. The pelvic X-rays are highly unreliable. The MRI of the SI joints is a reliable finding, is a useful finding, but it may also occur in healthy individuals, as we have seen in this particular study, which I did myself, in which we found 23% positive MRIs in essentially healthy participants, or in those who were complaining of chronic back pain, or in runners, or in ladies postpartum, etc., etc. It may also occur in patients without a particular disease. So what is the most important message for clinicians? Well, making a proper diagnosis of axial spondyloarthritis is properly recognizing the pattern of axial spondyloarthritis. You should build the picture, you should build the case. It takes some expertise, it needs some expertise. You should do more than only ticking a checkbox of criteria. That definitely doesn't suffice, but you need to recognize the pattern and to build the pattern. And in case of doubt, I would say, well, wait a while before you start a biological DMARD. So weigh the objective information of MRI and CRP. Do not rely too much on X-rays and MRI. And if an axial spondyloarthritis patient is brought into sustained remission, then reduce the dose. Do not stop immediately, but reduce it by half and see what happens. And if you cannot get an axial spondyloarthritis patient into remission, please reconsider the diagnosis, because I still believe that the majority, the large majority, maybe even more than 90% of the real axial spondyloarthritis patients are responsive to TNF inhibitors and or IL-17 inhibitors. So a note of caution is the following. This is one from a lady who's called Edna Ferber. She was an American novelist, maybe even a feminist in those days, in those early days of the previous century. And once she quoted, "Perhaps too much of everything is as bad as too little." I think that is applicable to the situation of axial spondyloarthritis too. Perhaps too much of axial spondyloarthritis is as bad as too little of axial spondyloarthritis. Thank you very much.